Firstly, before I actually go into the talk, I must thank the BCS Hertfordshire branch for inviting me to give this talk this evening. It's a real pleasure, and I absolutely mean this. I've been involved in what I used to call colour blindness since probably the late 1970s, a long time, as you will see as I go through my slides. So what I'm going to do is I'm going to relate on my experiences. So it's going to be experiential. And what I'm going to ask you to do is think about your experiences. When I start talking about colour blindness, or colour vision deficiency as we should call it today, people usually say something to me, I know somebody who's colourblind. Or it's, do you know, I'm colourblind. And then we get talking. And it's amazing the things that you find out. Later on in the talk, I'll give you some ideas of the statistics behind colour vision deficiency, how many people are colour vision deficient, and so on. But I'm not going to do it at the beginning. I'm going to be experiential. So here we go. Colour vision deficiency, is it a problem? And if so, whose problem is it? So what I'm going to try and do initially is to highlight that it is a problem. And then I'm going to end up, hopefully, by convincing you, especially if you're a software developer, that it is your problem too. Let's have a look. There you go. Is it a problem? Now, I don't have time in this talk to really go into the details of colour blindness. So this is the slide that Chris put up very kindly with my photo, a very youthful me. So I've got a lot grayer since then, with a picture of an eye in the background. It's lovely. Now what I have done, if you look top left-hand corner here, it says Deuteronopia. This is how somebody who suffers from Deuteronopia would see our original slide. I'll just bear with me a moment while I ask Chris a minor technical question. Can the cursor be seen wiggling around, Chris?
Yes, it can, Colin. Yes, it can.
Oh, thank you. So now what I'm going to do, here's another type of colour vision deficiency known as protanopia. And this is how people who suffer from protanopia would see the same slide, that one. And finally, there's only three that I'm really going into, three types. There's this one called tritanopia, which, as again, you can see the differences. Now let's just have a quick comparison. Initially, well, all we're going to do is look at my face. I'm very rosy, cheeky red there. I've lost all the redness here. I've lost all the redness here. So straight away, we can say deuteranopia and protanopia, we are losing red. However, we are not losing red with tritanopia. And what we can do is we can go through each of these. If we look here, we've got a nice purpley colour. Down there, it looks grey to me. I don't know to you, but that looks very grey. So what we are losing here, we're actually losing blue. Again, if we look just above where we've got the word colour and compare it with colour up here, a lovely blue there. Now it's a wishy-washy blue. And I'll go into things a little bit better as we go through. So there you are. That is how I would be seen by me, because I am not colour vision deficient. And even if I was colour vision deficient, that's still how I would see myself. But three different types of colour vision deficiency. So here we go. Some terms. Colour vision deficiency. It's really about not being able to define or see certain colours, hues and shades. Colour blindness, and this is why we don't tend to use the term today, that is the total inability of being able to differentiate any colour. So what that means is whoever suffers from colour blindness, all they can see are shades of black and white. It's extremely rare. And just to tag it on to the other three that we've looked at, Deuteronopia, Protanopia, and Tritanopia, this one is called monochromacy. Going into some more terms, just to really bring us into what's going on. I am not colour vision deficient. So what I am, excuse me, I am what's known as trichromacy. I can see three, what, chrome, colours. So I can differentiate between the three primary colours of red, green and blue. Dichromacy, this is where we're talking about the colour vision deficient in particular. There's only two primary colours that these people can actually differentiate between. Sorry, they have the inability to differentiate between. Got my words the wrong way around there. And they are either shades, hues, the colours red and green, or shades, hues, the colours of blue and yellow. If you think back to where I was with the initial slide of the images for this evening's talk, the blue and yellow is Tritanopia. The red and green is Protanopia and Deuteronopia. And there we go. Monochromacy, which I mentioned previously, that's black and white. Shades of black and white only. Rare. Just as a little injection here, it is a syndrome. No, sorry, it's a condition. I'm not going to call it a syndrome. It's a condition that is inherited. And if we go back to beyond modern day travel, you would find incidences or pockets in different geographical locations. Somewhere in the mountains in Spain, there is still a village or two villages or whatever neighbouring villages where people do tend to suffer from monochromacy. So with what we can actually say there is it's inherited in a local population, and today we have travel, so it's now spread around. Deuteronopia, well, I've mentioned that one. It's the inability to differentiate between reds and greens. So is Protanopia. You might ask me, well, hold on a minute, aren't they the same? And the answer is no. But what that comes down to is the biology of the eye. At the back of the eye, you have something called a fovea. And the fovea is the light receptor center, and it is full of cells. These cells are called rods and cones. And it's the differences between the rods and cones that show the differences between Deuteronopia and Protanopia. And also, if you think back, I did try to highlight the difference in that first slide where there was the four images. Tritanopia, that's quite straightforward. That's blues and yellows. Now, just to go back to a little bit about the biology of the eye, not much. These light receptor cells at the back of the eye, the rods and the cones, well, they are what, without going into detail, link into the optic nerve. So the signals are then passed through the optic nerve into the brain. And those of you who know about networks and mappings, that's where the mappings go, somewhere in the brain. And I'm not going into those details. We stop just at these terms. If anybody wants to talk about the biology, if I can remember it, we can pick that up later on. Here we go. This is the type of thing that we see. Deuteronopia. And we've got some lovely greens around here with a red core. This is how people with Deuteronopia would see the same. That green, I'm not sure how it comes across to you, but it's a murky, horrible colour. I wouldn't like to say what it is. And the nearest colour I could get, the red, would be khaki. But if you look at the contrast and the differentiation, I think you have to look closely to see them. If I had taken the outer edges off these cells, it may have been harder. I'm not sure. And this is Protanopia. So again, the same thing, green outside, red inside. This murky, whatever it is, no, that was khaki to me, I would say, with possibly a grey inner. I'm having troubles differentiating these two shades. I don't know about you. I'll leave that for you to decide. And now we have Tritanopia. Look at this. We have a blue with a yellow. Suddenly, the blue becomes mauve, I would say. And the yellow, light grey. I'll leave you to decide what colours you think they are. What we do know is that they are not what the non-colour vision deficient person would see. Oops, wrong button. Monochromacy, just to complete this, even though I'm not really talking about it. So there's our green with our red, and we have a grey with a grey, and I find that very hard to see. Perhaps you might see it easier than me. The only reason I know this box is here is because I can see the area that the red is taking and the position. I know exactly where it is. Now that's an interesting thing if we were to talk about traffic lights. As people say to me, traffic lights not a problem because of the red, amber, green. No, what we have there is not only colour, but you have spatial with a T difference. Here we also have colour with a spatial T difference. That's why I know that they're there. So colour and space, we can bring in another aspect and there isn't a problem. But as later on I'll tell you it's horses for courses. And this is how, oh, that's monochrome. I've said monochrome. Oh yeah, that's correct. So here we have the yellow, the blue with the yellow, and this is how somebody with monochromacy would see blue and yellow, dark grey, and that's a lighter grey inside the middle where the yellow should be, from what I can see. Me. Now, yes, this is something that Chris was very kindly picked up upon. My past, yes, it has been very varied. I've done a number of jobs, but from the CVD research, it's probably more like 20 years that I've been involved in CVD research on and off, but I'm only going to claim 10. I've stated already, I am not colour vision deficient, but I have bumped into many people who are either CVDt, colour vision deficient, or they know somebody who is. And I hope you have as well. Now, this talk, I did say I was going to try and do my best to make it experiential. So my life before I was a computer scientist, I was a life scientist. I worked in hospitals at one point as a microbiologist. Showing my age now, I used to work in a TB lab. It was actually in an era when the press was saying TB did not exist. Well, I worked in the northeast. I actually worked in Durham. And there are a lot of miners, or pit workers as we called them then. And it would TB is an associated disease of miners. And I worked very closely with the Pneumoconiosis board. Ouch. So I can't access any of the samples that I used to look at. So what I've done is I've downloaded one. And you can have a look at it from here if you wish. There's the website. And just to make sure that people are aware that TB is not just in your lung, this is from a breast. I do not know whether it's male or female. All I can tell you it's from the breast. Highly likely to be female. Now, the staining, a little bit of background. It used to take up to 6 weeks. Probably more like 3, but up to 6 weeks to grow tuberculosis on agar. It was a slow grower. Now that meant if we had a patient sample in Like this breast sample we're looking at here, this may be from a biopsy, and the patient may be under the knife, and we can turn around and say, yeah, I'll give her 6 weeks, keep the patient there, and then we'll tell you the result. You have to wait 6 weeks because that's when the only time I can call it negative. I might be able to call it positive earlier if you can hang around for at least 3, 4 or 5 or 6 weeks. Not really on, is it? Not. So what we used the microscope, and we used various coloured dyes. And the staining technique, these are called staining techniques, was called the Zeil-Nielsen. There is Zeil-Nielsen if you ever wish to look it up. And this is the background. See them lumpy things there? Well, to tell you what they are, they're pus cells. They're white blood cells, sort of dark things. These I don't care. That I don't care. Probably more connective tissue. But what I do care about, if you look where my arrow is, hopefully you can see it. There's a little red splodge. Or hopefully it's red. It's a little dash. That is a bacillus. A coccus, incidentally, is round. You may have heard of staphylococcus. So here again, I can see the the bacillus. Now I am particularly staining for tuberculosis. Or to be honest with you here, I'm not staining for tuberculosis. I'm staining for something called AFB, acid fast bacillus. And that's what I'm seeing here. I'm seeing an acid fast bacillus. Acid fast. It can resist acid. That's all it's saying. And that's one of the things about tuberculosis. There's a little cluster of them here. Now, let's pretend I have Deuteronopia. Now, I know where I'm looking to see the clusters because I've already pointed them out to you. And I thought, oh, sorry. We're not quite onto nefarious diseases. So hopefully you can see them. I don't know. I know I've got a trained eye and I did it for many years, so they stand out for me. They really stand out well. However, if I was Deuteronopia, no way would I be able to tell. I might be suspicious of that one there. But look at the colour of it. It's the same as the white blood cells. Deep blue. I look at Protanopia and it's much the same. And these are the red-green problematic people. And again, I can see that same little blob there. So I might be suspicious, but there again, I probably wouldn't be. And if I was a Tritanopia person, I might see that blob there, but I'm getting hints of the cluster up here. It's not easy. It's not good. It's very difficult. And this is where I really first, in my experiential world of meeting people who were, as we called them, colourblind, came across one. One of the chaps I worked with, he could never identify tuberculosis from the Zeil-Nielsen. Now, the idea of the Zeil-Nielsen was for me to turn around and say to the clinician, I have found acid-fast bacilli, which is indicative of, and at that point I would just have to say Mycobacteria species. By the way, that includes leprosy. Mycobacteria species. That's all I could say. But then that meant they could start the patient on immediate therapy or even isolation. Whereas this colleague I worked with, I don't know what type of colour vision deficiency he had. Probably we do know it was red-green, by the way, because we did further tests on him. So he was either Deuteronopia or Protanopia. He had no chance. And it took quite a while before the penny dropped that he was getting them wrong. And patients, let's go away from this breast sample here. Let's go to lung infections. So you actually, we called it sputa. Singular is sputum. So what is produced in the lungs? And we would look at the sputa, and it would look exactly the same as this once you've stained it. He did not pick them up. So potentially people in the northeast, in particular miners, working with the pneumoconiosis board, were walking around untreated. They could be infected their fellow colleagues, their fellow miners down the pit. Not good. So that was my first experiential practice. His name was David, by the way, but I stop at that point. What we had to do was, when he was doing the tuberculosis, someone else had to look at the slides. It was as simple as that. That was how we solved that problem. Yes, David, you can do the work in the TB lab so that you can get your qualifications. In those days, it was all institute qualifications, and we used to call it the special. Now it's called a master's, an MSc. So it enabled him to do it. And it enabled us to look at other samples, his samples under the microscope, so that patients were detected. We had a high success rate, by the way. Now I move on to me in the 1980s. And by this time, I've moved down to the southeast, where I am now. Well, I live in Welwyn Garden City, well, Hertfordshire. And this was actually at a hospital in St Albans, which is now closed. And what we were looking for was this brand new idea of using things called monoclonal antibodies, which you may have heard of on the telly, by this fantastic staining technique, immunofluorescence. And to be honest with you, it was a fantastic staining technique. I love it. Things that glow in the dark, things that fluoresce. And the idea of this one is, if we can have a look here, see this green blob, that green blob, that green blob, and that green blob, all of those green blobs, they're chlamydia. They are glowing in the dark. They are glowing bright green. And these red things that are attached, whoops, sorry, I shall go back, I'm too lively. These red things that are attached to the chlamydia, these are the antibodies, the monoclonal antibodies, and they are red. So you're now seeing the stuff that we're talking about with COVID in action, an antibody antigen reaction. So the chlamydia is the antigen, and the red stuff is the antibody, which was the monoclonal antibody, without going into any further detail. Now let's have a look how somebody who was Deuteronopia, are these actually shining in the dark? Are these smacking me in the face and saying, oh look, I'm fluorescing? And that's what I would be saying to anybody who is doing this technique. It must glow in the dark. It must smack you in the face. And you must see attachment, as we have here and here and here. So we can see attachment, but I don't think we see glowing in the dark. Over this one, this might be glowing in the dark. So this is Protanopia. So we now see a definite difference between Protanopia and Deuteranopia. But I'm not really convinced it's good enough to be as glowing in the dark as the green over here. And certainly while they get to Tritanopia, and this is the blue-yellow problem, no, we do not have glowing in the dark. And that means certainly somebody who suffers from Tritanopia, and certainly somebody who suffers from Deuteranopia, would have a lot of problems with this technique. And I also personally think people who are suffering from Protanopia would also have a problem. So that's me, the microbiologist. Eventually, I departed from the NHS and I got a job at the University of Hertfordshire, researching into neurotoxicology. And it was in the early days of growing neural networks. And we were looking at toxins on those networks. Today we would use stem cells. It's the forerunner of stem cells. And I actually taught this for about 4 or 5 years as a core module to the life scientists. So I've probably taught about 1,000 students this technique. And what it is if your cells are alive, then they can breathe. If your cells are dead, then they cannot breathe. So what we're going to do is we are going to stain the cell. And previously, I was just looking at the mass. Now I'm looking inside the cell. And we use this technique called Janus Green. I've got it from a Wikipedia site, this one. And the idea is you look, oh, drats, sorry, I'm being too lively with this. The idea is if a cell has respiration, then it will come up green. And what we're actually looking at are inside the cell, wee beasties called mitochondria. So this is mitochondrial respiration you're looking at here, and that is good mitochondrial respiration. Over here, if you have Deuteronopia, I'm not sure. I'm not sure whether these people would be able to see it. Now, what I'm going to do is I'm going to ask you to hold that number in your head. 1,000 students that I have likely taught this to. It's a nice easy number. I deliberately picked it, by the way. Let's move on. Okay. All right. This is where you might do a little bit of work if you like. I'm not going to ask you to do it now, but I would ask you to do it sometime in the future. Let's have a look and bring it bang up to date to COVID-19. And this is one of the places that I go to get my information from, the European Centre for Disease Prevention and Control. And there's a website for you. And what they do is they show you weekly maps here of where you might expect. Hello, something just went wrong there. Oh, I know what I did. Bear with me. Where you might expect, so this is countries, locations within countries and so on, where you might expect pockets of COVID. And I like this website, one, because it's highly topical for our talk, but it's very, very informative. And something that until I started doing this talk, I hadn't appreciated. They actually have, well, I knew they did this. They have a web page that shows a CVD friendly image. So what I'd like you to do sometime in the future is go play with it and have a go at this yourself. But we don't have time for it tonight. So here we are, European Centre for Disease Prevention and Control. Copy the two images, the current images. I put XX here because I did mine on the 7th of January. So we've got the 14th and the 21st. We may have more updated information than I've got. And this is the colourblind friendly image. Also, I did the 7th of January. And what it will do is put your current date in if you do as I'm asking you to do. And there we are. This is what I received on the 7th of January. This is the ordinary, the map of Europe. There's some labels here telling you what all the different colours represent. The UK looks very grey to me, so I think, I don't know, I can't find it. It's going to be down there. I'm sure things have changed today from the 7th of January, and we can see all sorts of colours. Now it's these sort of colours I want to look at. My geography is rubbish, so I have no idea. I think it's Finland. I have no idea. Those people who studied geography, I had to stop. I wasn't allowed to study geography. I had a choice of geography or history at school. When showing my age, I did O levels and I chose history. So I'm lousy at geography. But let's just take this. I'm assuming, and let's pretend that I'm correct. It's Finland. Well, that is the map that I can see. The map that they show that's supposedly Colour Vision Deficient friendly. Again, let's go back to Finland. Sorry, I can't see the difference at all there. I don't know whether you can. And let's have a look here. Again, sorry about my geography, it's rubbish. Look at all this round there. This is the friendly map. I have no chance. Spooky, the legend has completely changed. I don't get it. This legend is really weird. Let's have a look at the UK. Yeah, we can separate the UK quite nicely. Oh, look. I'm assuming we're in, I'm assuming we're in Spain here, because that could be Cherbourg up there. Look, here and here, there's a difference. Forget it. They've had it. No chance. Absolutely no chance. And that is a Colour Vision Deficient friendly map on a website. Let's have a little play. And what you can do is, there are many Colour Vision Deficient or colourblind emulators on the internet. This is one that I chose, Cobbliss, to use because it's quite good. It gives quite a good number of options for you to play with. So what I've done here is I've given you some notes on how to use it. So what we want to do is get the images, whatever image you're putting through, choose dichromatic view. In other words, we can only see two colours. We don't have RGB. We either have red-green problem or blue-yellow. And just play around. You can put any image you're liking there, and I would ask you to do this in the future. So this is me giving you sort of instructions, giving you ideas. Do it with the ordinary map, then do it again with the Colour Vision Deficient friendly map. And let's have a look at them. This is what I got from mine on the 7th of January. I am not looking at their so-called Colour Vision Deficient friendly image. So I'm looking at the ordinary. So I'm calling it non-CVD friendly. Well, actually, it's not that bad, is it? When we go look at what the same areas we were looking at before, I can see differences here. I can just about work out differences here. I can really, if I squint hard, make out differences here. Oops, I can't really see that one. The only reason I know about it is because I cheated. I looked at the ordinary earlier on. So if I didn't have this ordinary, we would be in trouble. So this is how someone suffering from Deuteronopia would see this map here. And somebody with protanopia would see it like this. And if we compare the Deuteronopia with the protanopia, they're very similar, but look at this difference in the darker shade here. I'm not quite sure what colour that would be, whether it would be grey or a dark green. I have no idea. That is certainly looking more like khaki to me, whereas over here on ours, we've got red. Now, the other thing I can see on ours, I can see borders from the different countries or provinces or whatever. There must be provinces. I struggle to see borders here. I personally cannot see any borders there. You might be able to, but I cannot. And then we go to the protanopia, sorry, the tritanopia, which is the blue-yellow, and you can see it's very, very pretty. But, oh, look at that one, how it stands out. And yes, I would probably be able to see this. But if we look at the legend, hold on a minute. I need the legend to help me interpret. That looks the same as that to me. That looks the same as that to me. And so on. All these legends are supposed to be different. And yet, many of the legends seem the same. So, I might say, which legend am I looking at? Am I looking at that one? I'm looking at that one. I have no idea. So even though we can look at these maps and say, oh, look, we can notice the difference, where do they fit in the legend? I have no idea. And that is how the CVDt would see the same map that we're looking at. Now let's go play with the one that was supposedly friendly one. Here's the friendly one that we looked at earlier on, which I hope I suggested to it. It's not as friendly as it should be. Now I seem to remember we were looking at a splodge here. Nope. We were looking at a splodge about there. Nope. And I think we were looking at a splodge about here. Nope. And even if we were, the legend's not helping me. So Deuteronopia have massive problems. Protanopia, not surprisingly, the same. Same. The legend again, forget it. I can't see what's what. I know I've condensed these down, but if you actually run these images through Cobbliss and put them on full screen, you'll have the same problems as me. He says, keeping fingers crossed and hoping. And here we are, Tritanopia. No chance. Look at that. No chance. No chance. Oh, actually, whoops. It keeps, I'm too lively. I can see borders here. I can see borders here. I have to squint hard to see borders in this one. Or boundaries. Here we go. I'll be leaving things in your head now. Hopefully people listening to this and watching this are software engineers. Hopefully people listening to this and watching this use colour. Hopefully. Now, if you are those type of people, colour coding, colour shading, colour whatever, here's some stats for you. About 1 in 12, about 8% of males suffer from some form of CVDt. When I showed these slides to Chris a few days ago, he said, well, how many would you say are red-green and how many would you say are blue-yellow? And if we say that 8 in 100 males suffer from colour vision deficiency, I would say approximately 7 of those 8 are red-green colourblind, i.e. that is Protanopia and Deuteronopia, and 1% or 1 in 100 or one of those people would be blue-yellow Tritanopia. Incidentally, I did say this was an inherited disease earlier on. Females can suffer from the condition, and it's about 1 in 200, about 0.5%. Okay, so we know it's inherited. We can see the male-female link there. I've said geographic, or it used to be geographic, now it's worldwide. About half the people who suffer from colour vision deficiency have moderate to severe conditions. The way things are today, and have been ad infinitum for these people, I don't know if you can remember it, I certainly can. When I was at school, I think I was probably 8 or 9, I was shown some pretty pictures, and I had to say what was inside these pretty pictures, and there were numbers and patterns in those pretty pictures. These are called Ishihara colour deficiency plates, and there are 38 of them, and they go back to 1911, I think. So over 100 years they were first developed. They were first developed. And I remember having that done to me at school. I do not remember what happened to any of my pals if they were found to have problems with the Ishihara colour plates. And if they did, what help was offered to them? Probably nothing. Oh, dearie me, you're colourblind. Clear off. That's it. Get on with it. So people have had to cope on their own. So they come up with coping mechanisms to help them. That's if they've been picked up. The two cases that I talked about experientially, me in laboratories, no, both of those males were not tested in their schools. Why? I do not know, but I know they were not tested because it came as a shock to both of them to be told, which was at that time we said colourblind. They were colourblind. And no, you could no longer look at these images under the microscope. You could certainly prep everything, but you could not look at them. Or you could look at them afterwards to see if you could spot what we were seeing. So those chaps who I've just mentioned there, in my experiences, I would say had severe conditions without realising it. Currently, there's no cure for it. There's some offer of help, but it's pretty poor. And this is where I have a little bee on my bonnet. I can go down to Hyde Park on a Sunday morning, I can stand on a soapbox and I can spout away about the Equality Act. We could say, are people who are colour vision deficient considered by the Act to be disabled? And if so, how are they protected under the Act? And if you read the Act, it will tell you the definition of disability. And in my opinion, reading that definition of disability, the people who suffer from colour vision deficiency fall under it. However, buried much further in the Act, you have it probably in some appendices somewhere, and it is buried, I found some exclusions. And guess what? They don't actually call it CVD, but they call it red-green colour issues. And they are not protected. It's not considered a disability. So you as a software engineer may turn around and say, well, it ain't my problem. I'm not having to consider accessibility for these people. Or am I? There's been, just to add to this, there's been a number of cases that have gone to different tribunals to try and get the Act overturned. And as far as I'm aware, none have been successful. So again, that's armoury in the people who want to say, not my problem, not my problem. Certainly the colour vision deficient problem, but not mine. So that brings us back to the title of the talk. Colour vision deficiency, is it a problem? And if it is, whose problem is it? So, is it a problem? Hopefully, through my experiences and from what I've shown you this evening, I hope I have demonstrated, yes, it is a problem. And yes, it can have big problems. Think about what we have today. And this is where the software comes in. I've got my little Android machine sitting next to me. I can't remember what the size screen is. It's about 5.5 inches, I think, by about 2.5 inches wide. I do a lot of looking up of stuff on there. If I'm suddenly asked to interpret some data, or no, I'm going to stop that. I'm going to say some information in a graphical form that I'm looking at on my Android machine, and I am CVDt, forget it. I've got no chance. It's tiny. I need to blow it up. I need it massive. I need it on the wall. So whose problem is it? Me, I say I'm non-CVDt, but it's my problem as well. Because if I am trying to convey a message to somebody, then I want that message to get across. I do not believe, in my opinion, that I cannot exclude such high percentages of people, 8% of the males, just because they are not covered by the Equality Act. Me, morally, I would have to help them. I would have to do something. I would have to consider the different colour contrasts, the different shades, the different hues, the different colours. And for those who are software developers listening and watching this, I hope you do. If you don't, you should. Hopefully you will in the future. So, do I consider CVD, colour vision deficiency, to be a disability? Well, I think it's pretty obvious I do. But that's my opinion. It's not according to the Equality Act, remember that. Do you consider, that's you, CVD to be a disability? I think that's for you to make your own judgment. But what I will say is I'm now going to bring those 1,000 students back in that I taught toxicology for 4 or 5 years to. If we now just do a straight split, 50-50 male-female, then 8% of those, that 50%, that's 500 students, are likely not to be able to see the mitochondrial activity respiration. So 8% of 500 is about 40 students. Well, I put my hands up and I own up to it. To be honest with you, I didn't even consider it. So these were, when they did these practicals, they were assessed. So about 3 weeks later, I would get their lab books in with the lab report in it, and I would assess it. And I will hold my hand up to this and say, well, those students, they got it wrong. So I can't give them any marks. And what I've done is I have unjustly, and I hold my hands up to it, unjustly marked down 40 students in the 4 or 5 years who were probably, it wasn't their problem. They couldn't differentiate, they couldn't see the mitochondrial activity that all the other students could see. And if we look at the 0.5% females, there'll probably be about, again, 500. My brain's gone on numbers. I'll leave you to work it out. 1 in 1,000, no, 1 in 200, isn't it? So that's about 2.5. About 2.5 females. So we chop a female in half, and that latter half was the problem half. Was it my problem as the tutor? I hold my hands up and I apologise for those students, because it was. They should never have been allowed to look for mitochondrial activity. I expected them to look for mitochondrial activity. I never considered, are you colourblind? Nobody came up to me in all those 5 years and said, Colin, I'm colourblind. I've been picked up with the Ishihara colour plates at school. So this green Janssen thingamabob that you're asking me to do, I can't do it. Nobody came to me. Was it my problem? I think so now. And I do regret not considering those students. So that leads me to ask you. I've held my hand up and I said I made a big mistake. In laboratories, we made a big mistake with the guy called David for quite a while before we found he was red-green colourblind and couldn't look at the Zeal-Nielsen. So how many minors were infected? We hear this thing about the R number. I can go on about the R number. It's not actually called the R number. It's called an RT number because it's a rate. What is the rate of infection? What is the rate of spread of disease? What are the virulence factors? What are the risk factors? And they're different. I can go on and on and on. But I won't. Because what I'm asking you to do, if you ever, whoever you are, reading these slides or watching the YouTube or whatever, use colour in your future work, consider these people and consider what you can do to help them. There we go. Coming to the end. So we'll go back to the roundup, and there's our slides that we started off with to end up, which I'm going to briefly flash because these will be on the slides, so it'll go onto YouTube. There's some references there if you wish to look at it. There's the one from the NHS, one from Colourblind Awareness. Then you've got Coblis, the simulator or emulator as I prefer. There's the Equality Act. All you've got to do is download the Equality Act as a PDF and Control F disability. And within a minute, you will find the definition of disability. I'm guessing within a minute. And there, beneath that, if we see, is there's a .gov.uk tribunal decision. Not in my favour. And that was in Dorset, some colourblind person, Mr. Bessel, taking on the police. He lost, and that was in 2016. So I'm going to take you back to my roundup slide, and then I'm going to pass it back to Chris, so Chris can do whatever he likes.
Well, thank you very much, Colin. That was extremely insightful and gives us all a pause for thought as well. I think the first question, we are rapidly running out of time for our hour here, but the first and most important question I want to ask you is, if somebody watching this is concerned that the issues you raise affects them, they may have a colour vision deficiency. Are there any people, places, groups or resources that you recommend they should head to immediately?
The first thing to do is just to check themselves out. There's a number of what probably called colourblind testers on the internet that they can go to and test themselves out. That way they'll get a better idea if they've got a problem. If they can, I would actually say get hold of the Ishihara plates. They're on the internet as well. Sorry, I didn't think about giving a reference to those, but I could do that later if wanted. And have a look at
we can put them in the comments for the chat video.
Okay, and look at those patterns. Once you've done that and you are convinced that you have a problem, then go report it to your GP. That's the only thing I can consider you could do. However, the GP will tell you there's no cure.
Is it worth reporting it to your employer as well? Would that be a good recommendation?
Yes. Actually, thank you, Chris, for mentioning that. I think it's very important that you mention it to your employer. In particular, just think about the way we're bombarded with information these days and what we have to look at. The amount of histograms, for example, that we get thrown at us. Well, can I actually interpret the histogram? And if the answer's, I'm having difficulty, whose problem is it? Is it mine? Or is it my employer's? Or is it both of ours? So yeah, I think you should. I think it should be noted. Whether it goes, if you have an occupational health department, I would certainly say get it on their records so it's noted.
Good advice. You mentioned there's no cure, but are there any technical or mechanical tools or aids such as glasses, special camera rendering, colour space manipulation that can help somebody with the issue?
Yeah, there's two types. You mentioned glasses. Yes, there are. There are glasses available, but they're not very good. They're still in their infancy. And the glasses that are available are not tailored for a particular type of the condition. So they're not tailored for deuteranopia, protanopia, or tritanopia. They're just tailored for colour vision deficiency. So they are problematic. They're not as good as they're supposed to be. I tend to consider them, I don't know if people remember, the old polarized lenses that we had in the 80s and 90s for sunglasses. They sort of work in a similar way to those. They try to correct it, but it's a bit wishy-washy. Now, as far as software is concerned, it's horses for courses. There are software available that could help, and the biggest form of research is into, or the most common form of research, into what's known as transformation algorithms. you mind if I get a little technical here? Probably what I would consider O-level physics.
Go for it.
1972, was it 73? Anyway, a long time ago. You have to consider the light spectrum. So we have a spectrum of colours, which mean these light spectrum, we have different wavelengths, lambda. And we have high wavelengths, high lambda, medium wavelengths, and low wavelengths. So these are the lambda. And that's where the colours fall. And these transformation algorithms, all they do is change something from, say, let's say high, one colour, to low, or from medium to high, whatever. And what I found with my experience with transformation algorithms, they have knock-on effects. What we did when I worked at the University of Hertfordshire, we developed, and we've had some success with it. Certainly got my last PhD student through. I would have loved to have done lots of more work, but wasn't able to. Was we developed what we called a colour separation algorithm, and what that did was we took an image, and where we saw different colours, so we went through it row by row, pixel by pixel, each image, and where we differentiated two colours through the goodness me, I forgot to call it, through the RGB matrix, then we simply inserted a white space. So it's a white pixel. And if you think about this, if you're scanning row by row by row by row, then you will actually build up a number of white spaces at what we call the colour interface boundary. So we separated the two colours. And then once it was completed, it was called the halo effect. And we had a good measure of success with that. However, as with everything else, it's limited in its use, because that's fine where you have big masses of colours, such as the maps that we were looking at. It would be ideal for those maps. But where you have lots of other things, the legends, for example, where you might have a word, so you've got difference in colours immediately, it would become very pixelated. So we had to put in things to allow for legends and things like that. It's not 100% successful, but it helps. And we ran a number of studies on human beings. We couldn't actually use real colour vision deficient people from clinics and so on because of the nature of the studies and the ethics. So for ethical approval, we just used a diverse population of people, students and staff at the University of Hertfordshire. And for each study, there's probably over 200 people per study. And we looked at a number of ways and a number of patterns and things like that. And we came up with some really interesting results, such that, well, Majid Al-Safiani, who did this work, who's now returned to Saudi Arabia, where he's from, was awarded a PhD for it in December last year. And he came up with some interesting things. So something quite simple that people could do, other than think of the colour transformation, change the colours and see what happens, is, well, why don't I whack a white space in between things? Especially things like pie charts, histograms, maps, white space. The white space is ideal. So that's what we came up with. I would really have liked to have done this through clinical trials, but we were unable to for a number of reasons.
It's very interesting. Well, unfortunately, we are extremely pressed for time.
Oh, sorry.
Just looking at the YouTube live comments, Ian Peel suggests that if you have the issue, you should let your occupational health team know if your organisation has one. And very quick, in a few words as possible, JMG wonders, should you report having colour blindness to your insurer, for example, your car insurance provider?
No, I don't think you need to there. The main thing, if you remember when I was talking about traffic lights, I said you have spatial difference as well. So when you're driving, the likelihood is you'll have spatial difference. So for example, if we're looking at a triangle warning sign for perhaps say a give way, it makes no difference to us. We know it's a triangle, we know it's a warning. Whether the words are in there, give way or not, doesn't matter. We know. We've got space there as well. So that one, I would say, not particularly. But that's me, personally, giving an opinion.
Okie dokie. Well, we'll have to leave it there. If anyone has any other questions, please do tweet them at us or email them in via the website, and we will get the answers out via Twitter or through the comments in the chat. So it just leaves me to thank Dr. Colin Egan for his time and insights this evening. It's been very interesting, and certainly from a software developer, a lot of pause for thoughts as well. So that just leaves me with enough time to say to you all, have a good evening. I hope you'll be able to join us on Tuesday the 16th of February, when we will be joined by Ken Munro, a partner from Pentest Partners. He is a media pundit and something of a white hat hacker, and he will be talking about hacking transport systems, including and not limiting to maybe ships. So please join us at 7 PM on Tuesday the 16th of February. Like, comment and subscribe, and don't forget to hit that subscribe button to show us your support for the BCS Hearts channel. On behalf of myself, Chris Tilley, and the committee of BCS Hertfordshire, we wish you all a very pleasant evening.